much, sir. All the best for your future endeavors, and we would look
Thank you,
forward to listening to you always.
Thank
Thank
you.
you so much, sir. So I would next like to invite Dr. Poornachandra B. He is an accomplished ophthalmologist. He has completed his PG from AFMC, and after that he has done his fellowship from VR in Narayan Nethralaya. And his area of interest is inherited retinal diseases, retinal vascular disease, and gene therapy, for which he has done his fellowship from Moorfields, London. So welcome, sir. Looking forward to your talk.
Thank you, Nilesh, and I would like to thank all the core members of OC for giving me this opportunity, and also thank you for accommodating my talk little early. So I'll be talking about the basics of gene therapy. I know there are a lot of postgraduates and residents who will be listening to this talk. So I thought I'll cover very basic aspects of gene therapy. So before going to the gene therapy, you need to understand what is gene. It is the study of heredity in general and of genes in particular, and it forms one of the central pillars of biology and overlaps with other areas like agriculture, medicine, biotechnology, etc. And we all know field of genetics is evolving at a very high pace, and it's very surprising to know that eye is the second only to the brain as an individual organ in its frequency of involvement with genetic disorders. And almost 50% of pediatric blindness will have some genetic etiology. And clinical picture also can be overlapping sometimes, which may not be enough for proper and full diagnosis. So before going to the gene therapy, I'll just show this example just to reiterate the fact that gene therapy is the need of the hour for some group of patients. So this was a 16-year-old female who presented to us in 2017. If you can see the autofluorescence, there is some definitely atrophic changes in the foveolar area, which progressed over next 2 years. Atrophy became even more prominent. By 2021, so there was a total foveal atrophy. So now she's 20 years. On presentation, she had a vision of around 612. Now it's 660. So this was her OCT and presentation in 2017. Over 4 years, this complete atrophy. So basically, this is a very mild variant of Stargardt's, which is usually Stargardt's happen because of a defect in a gene called BCA4. If at all, if you want to progress this disease from an initial stage to an advanced atrophic stage, as we saw in our case, what we need to do is we need to fix this gene called ABCA4, which is defective. So that's where the role of gene therapy comes. Basically, gene therapy, it is a technique of correcting effects of a defective gene that are responsible for the disease development. There are different approaches by which it can be done, where a normal gene is inserted into the cell to compensate for a non-functional gene, or abnormal gene can be traded with a normal gene, or we can also do an abnormal gene can be repaired without replacing. We can just repair that gene by a technique called selective reverse mutation or gene editing. And sometimes we can introduce a stop codon. There are some exons which are usually the coding sequence. By introducing stop, so we can do some kind of a gene editing. And sometimes what we can do is we can also intervene at the regulatory sites of a gene. We need not directly interact with the gene. By changing the regulatory sites also, we can, to some extent, we can give some functional recovery. So for this gene therapy, we use something called adeno-associated vectors. So these viral vectors are most preferred compared to other vectors. The reason being, these have got a very efficient delivery system, and they have got a very high level of gene expression, and they don't have unwanted this viral genomes which can cause undesirable immune response, toxic reactions in the host. All these are relatively almost none. Okay, and also most important thing is it has got an excellent stability, which allows it to be manufactured, stored, and handled so that it can be safely transferred to the delivery site. So that's a prerequisite for any new drug what to develop. From the manufacturing site to delivery, it should be stable without losing its efficacy. So these are the different gene therapy trials which are currently underway. I will not go into the details of each and every trial. So these are our main focus for gene therapy, because these are quite common in everywhere. Of course, in Indian setup also we keep seeing these cases quite often, like retinitis pigmentosa. LCS starts very early, Stargardt's, some of the corneal abnormalities. So, and we ophthalmologists are lucky because there are several factors which makes eye an ideal organ for gene replacement therapy as compared to the other organs in the body. The reason being, eye is easily accessible, and we all know it's immunologically privileged. And because of the small size, whatever we inject, either intravitreally or subretinal, it kind of get compartmentalized. So the systemic absorption is little less, so no undesired immune effects. And the very good thing is we have the possibility of using a contralateral eye as the control. Okay, and monitoring is also easy. We have many non-invasive monitoring like OCT, autofluorescence, ERG, where it becomes very easy. The effects can be seen and monitored easily. I'll just show a short video which I made. It's a kind of a cartoon which I made last maybe 2-3 years back, which basically shows the how a gene. Basically, it's a form of treatment that is offered to people with single gene defect. This is very important. For multiple gene mutation, if it's causing a particular phenotype, then it becomes quite complex to correct it. So preferably we prefer patient with single gene defects. So where a virus is made, what you call virus DNA is taken out and replaced with the one that treats the patient. And this virus is mixed with the normal human cells and they are cultivated. Then they are injected into the, for the patient for the treatment purpose. Okay, I'll just mute this. So then they are injected into the retina. Okay. So this is the video which I took when I was in the Moore fields. So before doing the gene therapy, we do a complete thorough vitrectomy to remove all the residual vitreous. Then we select the site, and most preferred site is subretinal. This is the subretinal injection which is being done in a case of choroideremia. Okay, so once this is done, these viral vectors will go and bind to the plasma membrane. They get internalized and travel all the way. The virion particle uncoats itself, it releases the material, and this will travel all the way to the nucleus. Once it is inside the nucleus, it will get incorporated into the host genome. So where it is, this is the animation showing the incorporation. So once this is done, it is expected to function normally at the way we have edited that gene. Okay, so that is in brief or in simple. It's not that simple a process, it's quite complex. This is the thing, this is how it is done. This was the team in which I worked. Team about initial when I was getting trained for gene therapy in more fields. So, and mentioned like see, these AAV vectors, they have many advantages, but there are some limitations also. For example, if you take our case example which happened because of the ABCA4 gene, this is quite a large gene, but the cargo capacity of AAV vectors is quite limited. So because of which we have come up with a novel technique called dual vector technology, where we split the gene, they are loaded into the separate variants, and they are co-infected together. Once they reach the host cell, function normally. So this is the these are the scientists like Professor Arkand, Shomi Bhattacharya, who are the main. Is their brainchild. This is the dual vector strategy. This is an ongoing research in Narayan Nethralaya. So why there is an unmet need for gene therapy in India? Because we know the patient, there are many patients of retinal dystrophy. We are fed up of telling there is no treatment and all that. And most of the disease, they occur in early childhood. And when we diagnose a retinal dystrophy and when we diagnose patient, but also for the parents and the family. And we all know, like a couple of years back, FDA approved only one gene therapy product called Luxturna, which costs around 6.5 crores. Obviously, Novartis is trying to bring it to India, reduce the cost, but still, I mean, 99.9% of Indians can't afford this kind of a therapy. But that's the reason the treatment needs to be developed indigenously to limit the cost to the patient. So once we develop the viral vector, first we need to do an animal trial. This is what I'm doing here. This is the injected some few viral vectors subretinally, and we also used some fluorescent bead tracking technique to make sure that the viral vectors are in the correct place. So we have a dedicated gene therapy production platform laboratory with an expanding team of scientists. I was telling vectors form the vital role in gene therapy, but how to produce these vectors? So these vectors are produced in an area called, this requires class 100 clean room facility. So the class 100 clean room facility means basically it's a what is what we routinely use for cataract and retina surgeries are class 100. So these vector production facility are there are 10 times more sterile than our routine OTs. So after crossing several barriers, finally you enter the vector production facility. That's where the clinical grade vectors are produced. So we have built the first in India gene therapy production, vector production facility at Narayan Nethralaya. And also the national guidelines for gene therapy development and clinical trials is out in the ICMR website. Whoever is interested, they can go to the ICMR website can download these guidelines. In case if some of you are interested in starting these trials and all that, you need to go through these guidelines, get an approval from there is one committee in ICMR called Gene Therapy Approval and Accreditation Committee. You need to get an approval from them before you start any trial, either animals or humans. So if any of you are interested in knowing more and want to collaborate with, you're free to do so. So thank you so much.